Today's speaker, Emma Bodley. Emma, I'm just going to change the presentation to you, and you will now need to accept that to be sharing your screen. Emma is a botanical records and plant conservation specialist at the Botanical Gardens in Auckland. That's in the North Island of New Zealand. Right, Emma, if you're all set to go, you should be able to begin your presentation.
Cool, thanks Renee. Kia ora everyone, thanks for the introduction Renee. It's lovely to have you all join for my talk today. So I work at the Auckland Botanic Gardens, and today I'm going to give you a bit of a talk on how we manage ex-situ plant conservation in the face of plant pathogens. It's a pretty hot topic, and what I'm going to give you is some considerations for when you're thinking about plant conservation, the types of things that we as a provider might like to know, and some of the learnings that we've had along the way. So this talk is based on a paper that me and my colleague Beck Stanley have written for the Australian Plant Conservation Journal last year. So if you want to read more about it, feel free to check out that link down below. So if you haven't been to the Botanic Gardens in Auckland, we're a 64-hectare site in South Auckland, so we've got a bit of a South Pacific feel going on. And the unique thing about our Botanic Gardens is that we don't collect just one of everything. We have plants displayed that do well in Auckland conditions, and that's because we want to give our community ideas and plants that have robust information behind them as successful plants for their home gardens. So that's really important to us. As well as that, we also have 3 key goals, which are education, research and conservation. And these 3 goals are goals of all botanic gardens globally. And today I'm going to really just touch on plant conservation, although the other 2 are aligned to some of the things I'll talk about. We're going to focus on plant conservation. So at Auckland Botanic Gardens, we've done a lot of plant conservation over the years, and in the last few years, what I've been involved in is lots of things to do with seed banking and seed storage, seed collection from the wild, creating seed orchards, propagating and understanding how to propagate plants. It's often one of the challenges of growing threatened plants is learning how to grow them and what conditions they need, not only in a garden setting, but also when you transplant them back to the wild. We also try to help overcome some barriers for plants in terms of pollination. So the photograph here is of Rata-moihau or Myrtaceae bartlettii, and this is said to be self-incompatible, so it isn't able to produce viable seed on its own. It needs to cross with another individual. So some of the work we do is around hand pollinating and trying to produce viable seed, propagating plants for duplicate collections, but also returning them to the wild is the end game for us. The threatened plants that we work on, a number of them have lots of threats already. So Rata-moihau is one we've been working on for about 5 years now. So it's in the Myrtaceae family, so it's threatened by Myrtle Rust. However, when we received material of these plants, we didn't have Myrtle Rust to think about. We just had our kind of regular considerations to think about. So in the wild, there's only 12 individuals left, and they are threatened by possum browsing. So we received material. Usually threatened plants come to us really quickly. There's not a lot of planning around them. Someone might be doing a survey and realise they've declined really quickly, and they just act quick and get some material into a botanic garden to grow on. But in an ideal world, we do a little bit of planning before we receive material, and some key considerations for us is what's the time commitment involved in having these plants? How long are we going to be growing them? What is the kind of space requirements you're going to need? Obviously, when you take a cutting, it's a small plant, and as they grow up, they get bigger and bigger, and then they're going to take up more space in our nursery. We also need to think about how many plants we're going to end up with. Watering requirements, do they need daily, twice daily, once a week watering? And what's the purpose of the collection when it comes to us? Is it just for propagation? Is it to be a backup ex-situ population? And how are we going to transition that ex-situ conservation over to in-situ conservation? So sometimes I like to think of a botanic garden as a hospital where plants come to us when they're either sick, unwell, or their habitat isn't suitable for them to grow. And we care for them and look after them until those things are resolved, and then they can go back to the wild. So we don't stay in hospital forever, or at least that's not the goal. You eventually go home, and that's what we like to do with plants. But over the last few years, plant pathogens really have become an issue, and that is then adding another layer of complexity into plant conservation about how we deal with different threats, whether that's coated dieback, Myrtle Rust, or any future pathogens. So nursery biosecurity is a really important part of botanic gardens. And the nursery trade has, in the last few years, had a lot of research done into how they have been contributing to the spread of plant pathogens. Nurseries can spread diseases into wild ecosystems when a person buys a plant from a garden centre and they plant it in their garden and their garden backs onto a forest. It's not very far for that pathogen then to spread from the pot that it was originally in into the wild. And we've seen some recent work from Beecham in 2017 that concluded coated dieback almost certainly was introduced into 4 of those coated forest sites from contaminated seedlings that were raised in the Waipoa forest, which is a pretty scary thought. The nursery trade industry is also shown to move Phytophthora remorum, and this is a really great example of just 2 nurseries that had Phytophthora remorum in their nursery, and then they distributed plants across America, and you can really see how massive that implication is, spreading from the west coast all the way over to the east coast just from one nursery. That's many states that now have that strain of remorum just from them. So that's really led to a change in the nursery community, specifically in New Zealand. They're working really hard to reduce their pathway as a risk for plant pathogens. And we've been involved in the New Zealand Plant Producers Incorporated scheme, where they've designed a accreditation scheme so that nurseries can opt in and upgrade, upskill their nursery and their nursery staff so that they are mitigating as many risks as possible to their nursery. So the Auckland Botanic Gardens nursery provides 65,000 plants a year to Auckland regional parks through the vegetation program. So we're a part of this scheme that has been accredited, and therefore we can be assured that the practices and the protocols we're doing are up to scratch to ensure that we're sending out the most clean plants possible. Prior to the New Zealand system being implemented, we were part of an Australian accreditation. And one of the really good things about both of these schemes is not that it is prescriptive and it tells you exactly what you must do to become accredited, and gives you standards that you need to achieve, and that allows you to work over time to meet those standards, but also with budget constraints and technology constraints, you can work around some of the own individual challenges you might need. So we have recently published a paper in the Cibolia journal about what exactly we've done in our nursery over the last 7 years to meet the biosecurity protocols, if you're interested. So one of the challenges in a garden is growing susceptible species together, and how do we manage the existing collections we already have if they get Myrtle Rust? And this slide shows you a number of examples of Myrtle Rust finds we've had at the gardens, both in the garden and in the nursery. And one example we have in the middle there, the second one from the left, that's a Lophomertus cultivar. So when we got Myrtle Rust in 2017 and it hit all of the red dragons in the gardens, Not that it's a particularly beautiful plant, but we thought, let's do a trial of all Lophomertus cultivars to see if any of them are Myrtle Rust resistant. So we sourced all the cultivars and we started growing them in the nursery. They were under the fungicide regime that we were meant to follow, and unfortunately they all got Myrtle Rust. And they were in our nursery, so they were in close proximity to many threatened plants that we're growing for conservation, but also our revegetation plants. So that really raised some questions for us around when do we say yes to projects and what are the risks to bringing other material in. Another thing for us is that Myrtle Rust is still an unwanted organism in New Zealand, so you can't knowingly distribute a plant that has been infected. And this is a real, raises a conversation around ex-situ conservation about where is the best place to grow threatened plants? Is it sometimes best to grow them closer to the place where they might be repatriated to, as opposed to growing them in a region outside of there and then having to consider distributing them across New Zealand? So it kind of leads me on to how do you choose an ex-situ provider? There's a couple of key things, and it's an important decision to make before you embark on a threatened plant project. Firstly, what is your goal? What are you doing with that threatened plant? Is it for, are you wanting to grow it for a seed orchard? Are you wanting to get seed to seed bank it? Do you want to tissue culture it because it doesn't store well as seed? And these are all really good questions to ask of the provider you approach, because not all providers, botanic gardens or other nurseries might be able to accommodate those things. They might not have the right resources to support that work. Ideally, highly susceptible species are grown outside of the climatic range of that pathogen. So for example, Myrtle Rust, perhaps that Lophomertus project would be better suited in somewhere like Invercargill, where there is no Myrtle Rust there and it's unlikely to ever establish there. Another advantage of working with a botanic garden is that we are part of a network called BGANS, which is Botanic Gardens Australia and New Zealand. And we work really closely together on a number of different projects. So we could have multiple providers for a project. That's not necessarily one provider has to do everything. It's sometimes good to use all the resources of that network. It's also really valuable for having duplicate collections. And a great example of this is when we realised Lophomertus ballata was highly susceptible to Myrtle Rust. We thought maybe we should start a seed orchard. So a seed orchard is having lots of plants of genetically different individuals in one place where they'll produce seed, and then you can use the seed for either storing or for growing on for conservation projects. But most of our Lophomertus ballata got Myrtle Rust pretty quickly, and they really struggled in the gardens, and that raised the question of, is Auckland the best place to grow the seed orchard? Perhaps it's further south. I think it's a really good thing to point out that conservation of some of these highly threatened plants in the face of pathogens might not be in the region where they're from. There might be better places to do this outside of that climatic region. So we may have threatened plants and we need to deal with pathogen management. Providers of ex-situ conservation need to have a plan, really. They need to have thought about what impacts the pathogen might have on growing the plant, on their day-to-day management of that environment. And that could be things to do with resources, either financial resources, staff time, infrastructure that might be required to be put in place. And a good example of that is our Rātamoi Hau project. As I mentioned, when we received that material, we didn't have Myrtle Rust in New Zealand, so it wasn't something we had thought about. We knew it was coming to New Zealand, and so we preemptively put those plants into a glass house that is completely covered, so we can close it. It's one that our staff don't often enter, so we're minimising the potential for staff to be a vector for Myrtle Rust. But that then creates some extra challenges around being in a closed space where humidity is going to increase. That house didn't happen to have any automatic irrigation, so it required some extra staff investment in terms of hand watering. Taking on plants that require some kind of fungicide regime is also really challenging in terms of it's been costly, the staff time, but also it's an ongoing and definite commitment when you hold a group of plants that are so threatened, and legally we need to follow a fungicide regime. We're not able to stop that once we start, so that's a really big commitment and something we need to think about. Watering is a really big one, actually. A nursery environment is a great place for pests and diseases of plants. That's mainly to do with our irrigation. We're constantly irrigating plants, and usually, and at least in our nursery, a lot of our irrigation is above the plants, so it's not from below. So that creates moisture and humidity, which again is a great environment for Myrtle Rust. And in a house where there is no irrigation, and then we go through a COVID lockdown where no one can be on site, there's another challenge there that we didn't anticipate, where we then needed to get staff on site to water these very threatened plants so that we didn't risk losing them. So sometimes crisis management also applies to plants, not just people. An interesting thing around a lot of these threatened plants, and also plants in the Myrtaceae family, are that they're no longer commercially viable to grow. A lot of commercial nurseries just don't want to grow them because of the commitment that changing their watering system, doing the fungicide regime every couple of weeks. So they have decided not to grow many of those Myrtaceae plants, and that puts pressure on people like researchers, for example, who are trying to source those plants for research. But now there's a demand, but they're completely inaccessible. So we get requests quite a lot for plants to be grown for research, and that's another consideration for us, is can we have the capacity but also take the risk of growing highly susceptible plants for research when we already have collections of plants that we are conserving. So it kind of leads me nicely on to have an incursion response plan. It's a great tool to have if you've already agreed to that upfront prior to a incursion happening. There are lots of pathogens that we can anticipate we might get, and so we can start to think about those if they did arrive in New Zealand while we were working on a particular plant. And obviously a lot of this work takes a lot of time to talk with stakeholders, find out what they'd be comfortable with. Sometimes there could be plant losses. We might have a group of plants growing for conservation and they become heavily infested with Myrtle Rust, and we can't legally move those plants, so we'd have to dispose of them. And that could be really quite an emotional response to having those destroyed. There is a silver lining, and this is a good thing to think about in an incursion response, is if you did have an infection of conservation or propagation material, could those plants go to research? And we have done that. As long as you have, the researcher has the right permissions to move those plants, they could go to research and find out more about the pathogen, which is always really valuable. And that happened in the case of that Lophomertis trial with all the different cultivars I mentioned earlier. They didn't go to waste. They went to plant and food with research where they could be added to their kind of sentinel monitoring plot and learn more about what's going on with those. So it's not all doom and gloom if you do get an infestation, but it is really good to have considered what you'd do if it happened, especially because usually these responses need to happen really quickly. So if you've done the planning and the thinking ahead of time, it will save you time when it happens. So these are just a few of our recent publications which I've highlighted through today. So if you want to find out more, please have a read. Otherwise, I'm happy to take questions.
Thank you very much, Emma, for that excellent presentation. Now we have just one question so far, but I have a few of my own if we don't have too. The question here, do you think about how to manage native pathogens that are restricted to threaten native plants? And I think what the person means is the threat to the pathogen perhaps is potentially even greater in terms of its potential extinction. Is that something the garden incorporates into its thinking at all?
I'm not sure we have explored that extensively, but it is a really great question. And I think the only way I can answer that is to touch on the fact that we generally don't use chemicals on our plants. So we are only fungiciding our plants because we have to, but generally we want to preserve like the soil microbial community as much as possible so that they do have those native pathogens that are living in that state.
Yes, certainly a question of particular interest to microbiologists.
Yeah,
all right, a couple of very tricky, a few notes of my own now then. So when you use this term a seed orchard, you're referring to what might be otherwise known as a seed bank?
So a seed orchard is a little bit different. It's living plants, so you have lots of plants of different genetic individuals, and then the seed bank is what you take away from those plants. So we have a few seed orchards of things like Cucurbit, Clematis pernicious, which we just maintain, and every year we harvest about 10,000 seeds and store those in a seed bank.
Very good. And do you keep the seed bank there, or are you working in partnership with other organisations that manage that facility?
Yeah, we work with other organisations to store those particular seeds, but we do have some facilities, basic facilities really, if we wanted to store more for ourselves.
Very good. All right, another question has come in now from the audience. How long did the Phytophthora ramorum take to move from the west coast to the east coast of the US? Well, I guess that's going to depend on what you can remember from reading the paper. Can you remember?
I don't remember, to be honest, but I think all those details are in that paper. It would be worth checking it out.
Yep, I could look to put the paper in the follow-up email.
Yeah, good idea.
I'll just make a note about that. So paper, or I will entirely forget. Okay, so another question from myself, and I guess that kind of relates to the seed bank question, as you talked a little bit there about, okay, we need to figure out where else we might grow these threatened plants. How much then do you network with other gardens and organisations to facilitate that?
Yeah, we work really closely with lots of organisations. So we're very connected to botanic gardens across New Zealand. So if there was something that we couldn't grow and we had to make a decision straight away, I could usually just phone another garden. They'd be very happy to take on that project. So it's a nice community that we work in, and it is quite a global community. If people approached us for similar projects, we might be able to help them as well.
It's very good. And another thing that popped into my mind when you mentioned the big commitment that plant maintenance sometimes requires, well, if not you, then who? Who else in New Zealand? I mean, are you really the arc, kind of last hope for some of these plants? If you decide to say no, then who might pick up that can?
That's a really great question. Hopefully within our Botanic Garden network there is someone who could accommodate them, and I'm thinking kind of South Dunedin and Invercargill. Those are great gardens that have the same kind of resources that we have, so they might be able to accommodate those plants. And reciprocally, if there was something in the South Island that they couldn't hold, we might hold it up in Auckland. But we're always looking to extend that network, whether that's with commercial nurseries are often quite interested in helping participate in conservation, and also specialist growers. I know there's quite a few of those around that also want to participate. So it doesn't have to be the traditional botanic garden, but we are connected enough that we could find those people.
It's very good. So it's a little perhaps ad hoc in a sense, though, in
Yeah.
that way.
Yeah.
From a New Zealand Inc. perspective. All right, another question has come in. Do you think that buying seedlings or cuttings from nurseries might possibly spread Phytophthora even though they're certified, or even Myrtle Rust? So I guess perhaps this is about how much can you really mitigate these risks?
hmm I think you can be pretty confident if we've got an accredited nursery that you're unlikely to be spreading Phytophthora and Myrtle Rust. It's a bit easier for Myrtle Rust in that you can more quickly determine if your plant has symptoms of Myrtle Rust as opposed to Phytophthora. But the process behind the nursery, they do all sorts of things like ensuring their plants never touch soil. They sterilize their pots and things like that. The process is really robust, that they should be not spreading that around. And the nursery industry is really committed to not being part of the problem.
Excellent. And another question now that I guess perhaps gets back to my point of who's really doing what, and where is the oversight coming from? What is the role of DOC in these plant conservation efforts and projects?
Yeah, there's lots of room for different organisations to work together, and there'll be often these projects that certainly I've been involved in are usually iwi led, so they come to us with a plant that they'd like help with, but there's definitely, I think, some room for improvement in terms of having a really strategic national coordinated response to some of the threatened plants that we might not be doing just because someone else has come to us with something and it's not as threatened or as important as something else. So yeah, we've got lots of space to improve on that.
Yeah, excellent. Now at the the risk of getting too political, I don't think we're treading on too many toes yet, so we'll keep going. Kind of related, is there strong political support then for your work within Auckland Council?
Yeah, absolutely. Botanic Gardens is part of Auckland Council, and so they really support conservation work. And we also work with our other council colleagues on more local projects as well. So yeah.
Another tricky one, really. If a cultivar is designed for Rama Rama, well, will it be released into the wild if it proves to be more robust against the rust? I wonder who would even make that decision.
Yeah, I think that's a really tricky question to make, and I think it comes back to that kind of eco-sourcing debate about
That's right.
where things and where things come from, and I'm not sure if there's an answer yet. If we lost all Rama Rama, would we prefer to have something as opposed to nothing, potentially? A lot of threatened plants tend to be conserved in amenity horticulture, so in people's gardens. And I can think of a few examples like Ticamanthe speciosa, where there's only one left in the wild, but it's kind of been conserved because it's such a popular garden plant that it isn't lost. So maybe something like a Rama Rama cultivar in everyone's home garden might be the next best thing.
Yeah, it's certainly an intriguing thought, and I'm sure citizen science in terms of planning restoration projects or keeping an eye out on iNaturalist is one thing, but perhaps people would be quite excited to have the opportunity to actively conserve some native plants in their gardens. All
Yeah.
right, well, that's an excellent discussion there, following an enlightening talk, and we're at 11:30, so I think we can wrap it up now. And a video of today's webinar will be available on the Beyond Myrtle Rust website and will be emailed to everyone who registered in a couple of days' time. The next webinar will be Wednesday, May 13th. And we will be hearing from Anne Sawyer going international again with the webinar series. She is a molecular plant biologist at the University of Queensland in Australia. Talk is going to be quite different, I think, from what we've heard today, given it is titled RNA Interference Mediated Control of Myrtle Rust. I will see you all then, and thanks very much. Have a good day.